When a blood sample is analyzed for purposes of trying to determine possible impairment in a forensic context or therapeutic versus toxic monitoring in clinical pharmacology, the total amount of the drug in the bloodstream is that which is recorded, if the analysis is correct. However, the large question becomes, is this measurement a truly relevant measure? No. The plain and simple answer is no. Shocking as that may seem at first, the truth is that if you're trying to determine possible impairment or therapeutic versus toxic monitoring, then we need to understand the concept of, quote, free versus, quote, bound drug form. Otherwise, if we do not, we may misinterpret the pharmacodynamic effect based upon an analytical chemistry measurement. We need to examine drug distribution in conjunction with the concept of free versus bound drug form to form a completely intelligent picture that involves the concepts of pharmacokinetics, pharmacodynamics, and bioavailability. For any ingested drug, it initially enters the stomach, usually dissolves, and is then absorbed into the bloodstream. Next, the absorbed drug circulates in the vascular compartment. An equilibrium mixture of free and protein-bound forms is achieved as it courses through the body. What is of particular note is that drugs can bind to plasma proteins, usually albumin. While the binding of drugs is reversible, the binding affinity of drugs is variable. Certain drugs, which are weak acids, and hydrophobic drugs and highly polar drugs have the strongest affinity for binding to these proteins. If there are two or more drugs in the bloodstream, both with a high affinity to bind, they will actually fight and compete to bind against each other. Only unbound forms of the drug, otherwise known as free form of the drug, can act on a target site in the tissues and may cause a pharmacologically relevant effect. On the other hand, bound drugs are always pharmacologically inactive because they are not free to enter the tissue. The brain is the tissue of particular concern as that is the epicenter of impairment. The drug produces symptoms such as impairment depending strictly upon the tissue content. In a living person, we cannot sample the brain to get an accurate measure of the free form that is in that tissue, which is the most relevant part. In other words, we cannot measure blood content of the drug in the tissue of most import, which is the brain. Instead, we must measure what is in the bloodstream, which is both the free and the bound form. The blood sample testing by way of analytical chemistry cannot be. It is just a gross or aggregate measure of both the free and the bound form. So, as the analytical chemistry analysis of the blood measures both free and the bound form, and only the free form has the possibility of being pharmacologically active or possibly impairing, then we need to be particularly aware of the ratio that a drug has been shown to be between the free and the bound form to begin to possibly understand the possibility of impairment. Of particular concern are chronic users of drugs. For example, very nonpolar drugs, termed as fat or lipid-soluble drugs, are stored for varying intervals in fat tissue. Note, metabolites are always more polar than the parent drugs. As such, there's no such thing as a nonpolar metabolite. They can then later re-enter the blood from the fat tissue or release. If the concentration of drug and metabolite within the blood is high, as would be the case for a chronic drug user, then the equilibrium favors continued adipose or fat tissue storage. This makes later release into the bloodstream despite actual abstinence a possibility. Again, as an analytical chemistry result, as typically processed. Now there are some extra pre-analytical methods available that can be used to separate the free from the bound drug forms. However, these processes are infrequently performed in a crime lab. Contains a measure of both free and bound drug, and there's no distinction between the two. This release from fat tissue could easily be misinterpreted and provide for a false positive despite abstinence. A very important professor by the name of Marilyn Hustis has written extensively about this type of issue with marijuana. She and her colleagues have noted that their research, quote, exceptionally long detection times have been reported for cannabinoid metabolites in the urine of frequent drug users during abstinence. During the terminal elimination phase, an individual may produce consecutive specimens that test positive, negative, and then positive again over time, despite abstinence. Psychotropic drugs such as tranquilizers, antidepressants, deterring agents, etc. also create their own unique problems. These drugs affect users by binding at sites within the central nervous system, or the CNS. Because the CNS is quite remote from the blood, such psychotropic agents are frequently not suitable subjects for drug monitoring. So, in summary, an accused motorist who insists that he or she had not taken an impairing drug, yet who has a positive blood analysis for an impairing substance, and in particular marijuana or other very nonpolar drugs and nonpolar drug metabolites that may be stored in varying intervals in fat tissues, may be telling the truth. Due to all of this, they may be innocent.